So thank you all for joining us today for this NCCR Catalysis seminar with Professor Abigail Doyle from the University of California, Los Angeles on development of data science tools for synthetic chemists. My name is Francine Lagadec, and I'm program coordinator of the Swiss National Center of Competence in Research, NCCR Catalysis. And my colleague Lauren Gamp, the NCCR Catalysis Knowledge and Technology Transfer Officer, will give a short overview on the program, and then Dr. Teodoro Laino will introduce Professor Doyle. Her 45-minute talk will be followed by a 30-minute Q&A, and since she will be busy presenting, we kindly ask you to send your questions to me in the chat.
Great. So thank you, Francine. So for those of you not familiar, NCCR Catalysis is a Swiss-wide consortium funded in 2020 by the Swiss National Science Foundation and our home institutions, ETH Zurich and EPFL. Under the directorship of Professor Javier Perez Ramirez and co-director Professor Jerome Wasser, we focus on sustainable chemistry through catalysis and bring together 13 institutions, 46 principal investigators, and over 200 researchers, many of which we're happy to see today. Our research spans the whole catalysis landscape, from small molecule activation and the development of fine chemicals from renewable platforms to the tools we can use to analyze those catalysts and the implementation of them via scale-up and sustainability analysis. One of our 5 work packages focuses on digitalization, and we are pleased to dive further into that topic today. I would now like to pass it off to our co-organizer and one of our digitalization work package coordinators, Dr. Teodoro Laino.
Thank you, Lauren. Thank you, Marie-Francine. For me, it's really a great pleasure to host today Abby. We have meeting a few times in the last period for a certain number of events that are happening around digital chemistry, and I thought that it was a very nice occasion to have her and let us know more on her research lines in the space of digital chemistry. So I'll say just a few words. Abby is very popular, doesn't need too much presentation, but I think it's important also for young people to get inspirational role. So let me say that Abby, you started as an undergraduate at Harvard University, and then you did your graduation studies with a very prestigious fellowship, actually, from the Department of Defense, first in Stanford and then back in Harvard. In 2008, Abby was got actually an assistant professorship at Princeton universities, and in 2013 the professorship was actually confirmed with tenure as an associate professorship. 2017, fast forward, she got the Barton Hepburn Chair in chemistry, always at Princeton University. So you stayed in Princeton actually for quite some time until 2021, when you decide to move from the other side, opposite coast, California, University of California, where you are now appointed with the Solwiston Chair of Organic Chemistry, University of California. The number of awards are unlimited, so I'm stopping here just to avoid blushing, but it's really a pleasure. I hand over the stage to you, Abby, and looking forward to your talk and discussion afterwards.
Thank you, Teo, and thank you for the invitation. It's a pleasure to be here virtually and to get to tell you a little bit about our research. So I'm going to go ahead and share screen. Hopefully this will work well. Okay, just pull up the pointer. All right, and can you confirm that you see the big screen, not the slideshow?
Yes.
Okay. Great. So yeah, it's a pleasure to get a chance to talk with you all today, this afternoon, in your case, this morning in mine, in the area of data-driven synthetic chemistry. And I think as an introduction, it's really important to contextualize that my group is very much a group that's experimentalists. We do synthetic organic chemistry, and we're very interested in reaction development and catalyst design. And so really what that entails is, and I think this is broadly speaking what synthetic chemists are interested in, is connecting chemical structure to reactivity. And the questions that we often are faced with are things about how does changing a set of reaction conditions, how will it impact the reaction outcome, whether that's the yield of a reaction or the negative selectivity if we're thinking about changing a chiral catalyst structure, or sort of also very inherent in our thinking and our understanding is trying to understand how reaction structure or chemical structure relates to mechanism. And ideally, being able to sort of think in the inverse way about if we want to achieve a certain reaction based off of a certain mechanism, or how do we reason back to structure. And so I think a very classic way of doing this, but also one that's now very contemporary, is to do data-driven development. And when I say classic, I think as synthetic organic chemists, we think often of linear free energy relationships as a data-driven way of doing mechanistic work, such as the classic Hammett relationship or Bronsted equation. These are primarily have been used by synthetic chemists for mechanistic understanding, but there's early examples of doing this for prediction as well. But recently, we became interested in whether there are additional opportunities in the area of data-driven organic chemistry based off of the fact that we now have considerably larger access to data, experimental data, as well as sort of computational resources, and tools in the area of computer science have become much more accessible, including sort of coding languages like Python. And so the question became, could we take advantage of all of these developments in order to build tools that will allow us to do more with less data, or at least the same amount of data. And so I want to sort of walk you through 3 stories that are sort of asking this question of whether machine learning can help experimental chemists do more in the area first of reaction optimization. Then I'll talk a little bit about reaction prediction, and then the third topic will be in the area of mechanistic analysis. So reaction optimization is certainly ubiquitous in synthetic chemistry. I think both in academic situations as well as industry, where we're trying to search for sort of the global maximum in a very complex chemical landscape, wherein there are numerous variables to consider, both categorical like catalyst structure or ligand identity, as well as continuous like concentration, temperature, equivalence. And because this space is so large, right, if you think about all these dimensions for each of them, there are hundreds, if not more, sort of components that we could consider. We need tools as synthetic chemists to navigate this, right? We can't possibly run every experiment within the optimization search space that we're interested in. So unfortunately, in my lab, and I think sort of we're not atypical for an academic lab, the primary way that we undertake reaction optimization, especially when we're doing reaction discovery, is by looking at one factor at a time, right? So we'll say, all right, we're going to look first at ligand identity, and we're going to screen ligand identity, find the best ligand, then we'll switch to pre-catalyst or base identity. That's partly a function of the number of experiments that we can sort of run in our lab at any given point. If you have access to sort of technology to do sort of run more experiments, there is the opportunity of doing high throughput screening, wherein you can look at the combinatorics of the different variables. But even this can't possibly cover, right, that full space that we'd be interested in for an optimization campaign. And so, what is both more common in industry and is becoming more common these days is to use either design of experiments or machine learning based approaches to reaction optimization. And there have been a number of advances in this area. We're certainly not the first. However, many of them have been in the context of chemical engineering type problems, and therefore sort of are not particularly well suited to reaction discovery, where simultaneously looking at categorical as well as continuous variables, being able to run experiments in batches of not just one at a time, but 5 or 10 experiments at a time is important, as well as sort of other functionality that I'll mention along the way. So we became interested in this, and in particular in the idea of using Bayesian optimization for reaction optimization. Optimization problems are ubiquitous, not just in the chemistry area, but in many other disciplines. And in the computer science area, Bayesian optimization has emerged as one of the most effective at optimizing expensive to evaluate black box functions. And so just for those of you who are chemists, I'll give you a very brief background on Bayesian optimization. Let's consider that we're trying to optimize a two-dimensional space of temperature and concentration, where each of these black points could be an individual experiment that we want to run. And we're trying to optimize for some unknown objective. Let's say it's yield. And this is what the surface looks like, but we don't know that at the outset of our study. So in Bayesian optimization, we'd start with a collection of data points. I'm showing you 5 in red here that can be selected based off of various initialization schemes. We run those, we submit in this case, the yield outcomes to sort of a Bayesian optimization platform. And what happens is you build a response surface model that is meant to both allow you to sort of make predictions on the outcome at different temperature and concentration regimes, as well as to understand the uncertainty of the model, such that you can apply an acquisition function to select the next experiment. And there are a number of different acquisition functions. As you'll see, sort of the best that we've found when we sort of tune this on experimental data sets is one that nicely balances exploration, so areas of high uncertainty, and exploitation of data, so areas of high predicted mean. Then one runs that experiment, feeds that information back into the optimizer. The response surface model then changes, acquisition function tells you the next experiment to run, and you iterate, right? And you iterate until either you've found a satisfactory result, or it seems like the predictions and the uncertainties of the model have leveled off to suggest that there's sort of no better outcome in that search space that you've identified. So as a team, we decided to sort of tackle sort of developing code that uses Bayesian optimization for chemical reaction optimization. This is a great team led by a former postdoc in my group, Ben Shields, in collaboration with scientists at Bristol Myers Squibb, led by Jay Stevens, June Lee, and Jake Janey, and with a former colleague of mine, Ryan Adams, who's a professor in the computer science department, and his student Farhan. And so what we first did is we built this code really sort of with the goal of making it flexible for an organic chemist and a sort of reaction discovery. We tuned it on high throughput experimentation data sets that were in the literature. And then we wanted to sort of test it. And so that we could sort of test it where we had the ground truth, right, where we knew where the global optimum was in a chemical space, we decided to build a new HD data set. And so the data set that we sort of built was on this palladium catalyzed CH aerylation reaction, wherein one can vary a number of different parameters. We chose ligand, base, solvent, concentration, and temperature. So here are the various configurations for both categorical and continuous variables. And then for the ligand, in this case, it was monodentate phosphines. And these were chosen from the BMS library of phosphines, where we sort of mapped that chemical space and chose sort of 12 phosphines that sort of nicely spread that space. So this was approximately 1,700 experiments. And beyond just trying to see how the optimizer did in this chemical space, we wanted to compare it to how chemists might optimize the same reaction. So to do that, we built an online game, and basically the game asked chemists to optimize the same reaction within the same search space. In this case, we decided to do it 5 experiments at a time. So it asked the chemists who were participating, what 5 conditions would you first run? And then after you submitted those, the game would give you the results of the HTE for those 5 experiments and ask what next experiments would you run. And we had about 50 chemists play the game. So we also ran the optimizer 50 times. In the case of the optimizer, we initialized with random sort of 5 conditions. And so here's sort of what the outcome of this was. So what you're looking at here is yield on the y-axis, number of experiments, or number of batches of experiments on the x-axis. And it's sort of 50 runs. The dark black here is the average of those 50 runs. And what we were pleased to see is that within 15 batches of experiments, all 50 runs of the optimizer had found the sort of global optimum conditions, and in many sort of in much fewer experiments than even that. In comparing to the sort of human participants, there were a couple of interesting things that we noted. The first, which makes a lot of sense, is that the average yield of the first 5 experiments that the human participants chose were much higher than that of the optimizer. And I think that makes a lot of sense. The chemists who are participating had years of experience to help them select those first set of conditions that are likely to maximize the overall yield. Whereas the computer doesn't have any information at the outset of the optimization campaign. However, what you do see is that the rate of learning and improvement is much higher for the optimizer than the chemists. We also saw that the chemists, many of them, you can sort of see one example here, stopped early. We don't quite know why that's the case, right? It could be that they got to a relatively satisfactory answer and that was it. We're not sort of experts in game design, so we probably could have done better here. But it also could be that they thought that they had identified the highest yielding conditions. And this sort of points out an interesting artifact of this study, which was not something we designed. And that's that the best ligand for this reaction was one that had never been published for this type of reaction before. So it's possible that, right, the intuition that led human participants to get a good high yield on average in that first set of experiments also biased them away from exploring parts of this chemical space that were actually sort of most important for getting the global optimum yield here. So I think that points out this interesting sort of double edge of what we often describe as chemical intuition, right? It really can be to our advantage in many scenarios. However, it could also bias us in ways that are not effective, especially when we're doing reaction discovery. So after having sort of completed this stage of the project, I was pretty excited. I went to the lab, most of whom are experimentalists doing synthetic chemistry. Said, all right, let's use this, right? We're going to use it all the time now. But that's not what happened. And I think it sort of brought out current gaps in the functionality that we needed to address going forward. So for example, Will, who is a graduate student in the group who is developing this enantioselective aerylation of an epoxide using nickel photoredox catalysis, I suggested to him that he try using the Bayesian optimizer. However, right, in his case and in many cases in synthetic chemistry, he wasn't just optimizing for yield, right? He was optimizing for enantioselectivity, for regioselectivity in this epoxide coupling, for chemoselectivity, right? And that makes sense. Most optimization problems in chemistry are multi-objective. Whether that's some of these objectives, or it could be process mass intensity and more sustainability type metrics. And our current version of ADBO is only a single objective optimizer. Multi-objective optimization problems are very well known in other areas. The one or a big difference is that there's usually not a single optimum solution, unlike a single objective problem, but rather you need to optimize for a Pareto front. And so Will and Jose, a data X fellow in the group, decided to modify and sort of augment our current approach to optimization to include multi-objective optimization, as well as to include sort of many other functionalities that I'll sort of touch upon as we go. And just, briefly, that sort of required sort of adjustments in the response surface model and the acquisition function with the various HTE data set that we were able to get our hands on. We again tuned now the multi-objective optimizer, including sort of looking at different response surface models, acquisition functions, initialization methods, as well as in this case, sort of different ways of featurizing the categorical variables, such as ligand. And generally what we see sort of consistently is that using DFT typed features does outperform sort of alternative methods of featurization in these optimizers, especially at sort of later data points in the optimization campaign. However, you can see that there are a number of featurization methods that sort of work here, such that you don't need to sort of go to all the way to the extreme of featurizing by DFT to sort of make use of these tools. So after having sort of built the multi-objective version of our optimizer, wanted to see again how it would fare. And at this point, Will had actually finished developing his nickel photoredox method, but we thought we could use that then as a comparison to see how well and if the multi-objective optimizer could find similar sort of conditions as he had found manually. In his case, it took probably 6 months to optimize this reaction and approximately 500 experiments when we looked back at his lab notebook. And the chemical space that he had sort of covered in his optimization campaign, again, included both categorical and continuous variables. In particular, sort of the chiral ligand framework was absolutely critical for an anti-selectivity, but also things like nickel pre-catalyst, additives, solvents, light source, and concentration. So we used this as our chemical space for the evasion optimizer and looked at it for 2 different reactions. And I apologize, there should be a substituent on this aryl iodide since this is not a stereogenic center as drawn, but sort of believe me, it's a distinct aryl iodide there. And in sort of the case of his manual optimization, as I said, sort of in approximately 500 experiments, Will had found conditions sort of shown sort of where at the intersection of these 2 gray lines that delivered about 70% yield and 91% EE for this reaction. And then in blue are the experiments that our multi-objective optimizer, which we're calling EDVO plus, found. In the light blue are the early suggested experiments. The darker blue are the later suggested experiments in batch sizes of 3. So what we found is in a total of 24 experiments, the optimizer had found a slightly better set of conditions, albeit we were just happy that it had found similar performing conditions. So 80% yield and 91% EE, as opposed to the ones that were optimized manually. Interestingly, in this case, using the exact same chiral ligand, but different conditions otherwise. In particular, the nickel pre-catalyst concentration are different. We also looked at it for a slightly harder substrate. So this is a substrate that, using our standard conditions, gave 49% yield and 76% EE. And again, sort of in this case, within 15 experiments, the optimizer had found better conditions, about 10% yield increase and similar levels of an anti-selectivity. What's interesting is nearly every single sort of variable here is different between sort of what we had found and what the optimizer found. And when we looked back at the impact of each of these single sort of changes, what we see is that if you just change one of the variables independently, the yield or the EE decreases. So I think I have that on the next slide. Right, so if you point change each of these, what you see is that it leads to a worse result, even though the combination of all of them changed leads to a higher result. And so I think this again sort of speaks to the fact that, you know, chemical space is complicated, and that at least in academics, the way that we do optimization, typically using one factor at a time, is really not sufficient, right, to sort of identify sort of optimal conditions. Now, I think, you know, this was really exciting for us. You know, it still was the case that we were not getting most of my students in the group to use this. And so sort of the last thing that we've done here, although we'll continue to improve this, is that we've built a web app such that the students in my group, who I think are, you know, in general, similar to sort of many synthetic organic chemists who aren't that familiar with coding in Python, can use this tool. So we can move from the command line interface to a web app that's hosted on the cloud. So you don't have to install any software in order to use the optimizer. And we're hoping that now with this, both chemists in my group, as well as sort of chemists in academia, as well as industry, will be able to make use of some of these tools, and it'll be open source. We're just finalizing sort of and debugging a little bit, and it should be available very soon. Okay, so that's the story I wanted to tell on optimization. Now, there are many scenarios in synthetic chemistry in reaction discovery where we actually aren't interested in optimizing. It really doesn't matter whether we get 99% yield or we get 20% yield, as long as we get some material. And we want to be able to do so as quickly as possible and as few experiments as possible. And in this case, ideally, we would be able to use literature data to make predictions on you know, what set of conditions to use for a new substrate, or you know, how a new substrate will fare in terms of yield and EE, without sort of running a lot of experiments. And we can see how this could be useful in the context of things like medicinal chemistry, where you want to make a library of molecules quickly. You don't know which one, right, is going to have the desired functional, you know, bioactivity. So you don't really care if it's, you know, high yielding or low yielding, as long as you can get access to it. And you don't want to have to optimize. Likewise for multi-step synthesis and route vetting, as well as in reaction discovery, when we want to establish if a hypothesis is feasible, you know, we still need to sort of come up with the first set of conditions, right, to try. And so in these cases, we really rely on literature data to make predictions, either through intuition currently or through some of the advances recently in using machine learning for reaction prediction. However, I think has been described quite a bit recently. Published data and databases have certain limitations, right? They're incomplete when it comes to recording sort of the full set of conditions that were used for each experiment, things like concentration or equivalence, or sort of other variables that are important to reaction success may not be recorded. And also literature data tends to be biased. It's biased towards high performing conditions, right? It doesn't record failure very frequently. And it's also sort of biased towards sort of historical conditions rather than sort of, there are more examples of that than sort of more modern conditions. And so what we were curious about recently is not necessarily building a predictive, model, but what can we do as synthetic chemists who contribute to the literature to improve it, right? Are there ways that we can systematically change our practices such that the data that we're putting in to these databases will be more useful for data science applications? And what that brought us to is the idea of substrate scope tables, right? When we do reaction development, every paper that we publish is accompanied with a substrate scope table, and the purpose of that table is to demonstrate the generality of our new reaction conditions on a diverse set of substrates. However, the way that we do this in my group, which I don't think is too atypical of the community, is not systematic, right, in terms of how we select these diverse samples. It's typically based off of what we have around, what's easy to sort of access. We do try to vary substituents in ways that, we've got electron donating, withdrawing, some that are sterically hindered, but also because, we want to publish this, we do sort of, focus on success. There may be some limitations recorded, but perhaps not all of them. So, the question is, how can we do better? And is there ways in which we can sort of have a much more systematic approach to this? Because for sure, the way that we're doing this and then we're putting this into the database is not helpful in allowing chemists to do what they want with this, which is to be able to predict how an unseen substrate will perform, to understand what are the limitations in the chemical space. So in particular, so the question is, can we systematically choose a substrate scope that would lead to both success and failure, and it leads to a data set that would enable better predictive building of predictive models. And so to do this was actually, we're not innovating here, but borrowing from sort of chemoinformatics and library design. And the general workflow that a group of my students came up with is shown on this slide. So this is a project that was highly collaborative within the group from Stavros and Shutian, who were in fact developing a new synthetic method that we wanted to apply this to, alongside Andre, Shivani, and Jesus, who were sort of working on sort of data set design strategies. And the general approach is that we do sort of a reaxis search for all examples of a given substrate class. I'm going to show this to you in the context of aryl bromides, which delivers about 700,000 that are commercial. We then apply certain filters to limit based off of things like molecular weight and cost, so that these would be compounds that we could actually sort of use in a substrate scope campaign. We can also sort of limit based off of functional groups that are going to be incompatible with a given chemical reaction. And it's important then to record this so that it's available to sort of chemists and also in building predictive models. Then we need to featureize those molecules, and we use DFT featurization to do that, to capture the steric and electronic properties of the substrates that should connect to reactivity. We have compared a few different featurization approaches in this process, not exhaustively, but have found that DFT featurization outperforms Mordred descriptors. And then we need to, because that'll deliver 100 or so features for every given molecule, we need to do dimensionality reduction and clustering in order to select a substrate scope. And I'll just mention sort of for the featurization, DFT featurization, since we're dealing with thousands of different molecules here, we have built, Andre in particular, as well as Ben and Jason have built a high throughput feature generation sort of code. It's called AutoQChem. It's open source. And it allows us to take SMILES strings, then do sort of conformational search through RDKit, and then submit these to DFT featurization through, in our case, Gaussian calculations, and then store these in a database that's searchable, sort of as shown here, so that you can pull up, if you go to this site, all of the different DFT calculations that my group has done, and can download feature sets if you're interested in. So in this case, we did this for aryl bromides. After dimensionality reduction, using UMAP, we can sort of visualize the chemical space of aryl bromides. And in this case, those are all the gray dots. Because we were interested in applying this to a nickel photoredox method, we decided to search the literature for all papers, nickel photoredox papers that used aryl bromides as substrates. And I'm showing them in blue here, where the size of the dot corresponds to the average yield of that aryl bromide in the synthetic methods that it's been applied to. And even this visualization, I think, is really helpful, right? What you can immediately see is that the right-hand half of this chemical space is more populated than the left-hand half. And also that, right, higher yields tend to be localized onto that right-hand half. Yeah, what that you can even see, right, islands of aryl bromides where there's been no published examples. We don't know if that's because someone's tried it and it hasn't worked, or if no one has looked into that chemical space. But I think this points to some of the value of doing these visualization tools for reaction discovery. Just briefly, just point out a few examples of chemical structures superimposed here. What you tend to see is that monosubstituted, parasubstituted aryl bromides tend to sort of be over here. You can see that some of the more hindered substrates fall over on the left-hand half, and they tend to be on average lower yielding. And then in that island that I mentioned, these tend to be substrates that, as someone who is familiar with the area of nickel photoredox, I can say sort of, with some confidence, these are really challenging substrates. They're electronically and sterically deactivated. So my intuition would say that if someone has tried them, they've failed, and that may be why they're not represented in the literature. But we would like to know that, and we'd like them to be in the literature. Is certainly an important point here. So in this case, we then clustered this chemical space, and we chose the centermost molecule from each of 15 clusters to represent the substrate scope for a new method that we were interested in. Those 15 molecules are shown here. And I think the point to make here is that these, from just a visual inspection, are much more diverse than the ones that my group would typically choose if you asked us to choose 15 substrates for the same substrate scope manually. In particular, you see that there are many more multi-substituted aryl bromides than we would usually have. And because of that, you can represent many more functional groups in 15 molecules than you could if you were just looking at monosubstituted aryl bromides. And so in this case, Stavros and Shutian applied these 15 substrates to a new synthetic method that we're developing. The method itself is sort of not important for this talk, but it's an alkylation reaction of aryl bromides using these dialkyl acetals as radical sources. And when we did so, we saw a range of outcomes from very high performing 98% yield to very low performing 0% yield in these two cases shown, and everything in between. And what's beneficial about this is then we can sort of compare, our method to the literature and start to make some generalizations. And this could be helpful, for someone like a medicinal chemist who wants to choose among various methods to apply to an unseen substrate to try and figure out which of the methods, which of the conditions would be most effective. What we see sort of in this case is that the method seems to be better and more tolerant of hindered substrates than what's represented in the literature on average. I can see sort of a couple of cases here, but we do see, that the substrate that was in this cluster that's not represented in the literature gave a 0% yield. But this will be useful, to publish, and we have published that now. We also noticed a couple of cases where the substrate in an area where sort of the literature had high performers was not very high yielding. And we wondered if this was because, the substrate is representative of the method in that cluster, or in that there was actually sort of this method was less effective than other literature methods, or if there might be some other reason why this substrate was not a high performer. And so to just quickly look at this, we chose another 3 or so aryl bromides that are in the same cluster. And what we see is that they all give quite high yield, these 3, as opposed to this imidazole containing aryl bromide that was lower yielding. And so what this points out, and it's an important point, is that, the goal here and sort of the message is not that one substrate within the cluster is going to be fully representative of the reactivity of all the other substrates in the cluster, right? Because there are thousands of aryl bromides here, right? There are hundreds of aryl bromides in a given cluster. So you might ask, well, what's the point then of doing this? Well, the point is that if we can build a model with these 15 examples that is predictive of these other examples, performance, and it's a better model than a model that we built off of literature examples, then there's a lot of value in doing this approach, even if a single molecule is not representative of its cluster. So that was the next question we had, is can we build a model off of these 15 sort of diverse, if you will, aryl bromides that can generalize to unseen examples. And so in this case, we built really, really simple univariate model, not trying to, since we only have 15 data points. And what we found is the highest, the most predictive univariate feature was electronegativity of the aryl bromide. So using the 15 generalized additive model gave us a root mean squared error that is not, this is not sort of highly predictive of 20 on R squared of 0.56. And then the question is, how well does it generalize? To evaluate that, Stavros and Shutian ran about 35 other aryl bromides. What we found is, sorry, let me sort of fast forward here. The aryl bromides that are sort of the light blue triangles are the validation set. We see that it validates very well, right? So root mean squared error of 17, R squared of 0.64. So the regression model built on only 15 aryl bromides that are selected through this workflow captures 60% of the variance and describes the trend of yields for these validation aryl bromides, which I think is quite good, especially if you compare it to a similar model built on the literature examples. So the triangles that have the dark shading on them are examples that were in the literature. And so if instead we try and build a model on those literature examples, what we see is that the training error, right, is quite good, root mean squared error 14%, but the validation is not good. And that's because it's sort of hard to see here, but most of the examples in the literature are the high yielding ones. And so this model really can't predict all of the low reacting, the failed examples. So all of this to say that I think this illustrates some of the value of using a more systematic approach to selecting substrates for then what the outcome of the literature will be in building models. I'll point out as the last point here that this model is imperfect and this approach is not complete. In particular, what we see is while the model is actually very good at predicting examples of aryl bromides where substituents have what I'll call an internal effect on reactivity, right? So it's sort of either they're modifying the electronic or steric properties of the aryl bromides. The model is very good, but it's not very good at predicting external impacts of functional groups. And I'll point out this aldehyde, these two examples, right, that are aldehyde containing substrates. In these cases, right, an electronegativity model predicts that they should be quite high performing because aldehyde is an electron withdrawing group. But that aldehyde can actually serve as a poison through hydrogen atom transfer. And we can sort of see this if we do a glorious fragment type approach where we add in an aldehyde to a reaction of a substrate that's otherwise a very high performing reaction. We see that the reaction yield decreases and the aldehyde is consumed. And so right now, we're thinking about sort of in terms of moving forward, ways in which we can featureize aryl bromides and build models that can capture both internal and external effects of substrates. Because right now, the current approach that I've just shown only really captures the internal impact of functional groups on a substrate class. Okay, and then the last point, third story I want to tell in just the last 5 minutes or so is related to using sort of data-driven approaches to mechanistic analysis. This is probably the one that's most familiar to organic chemists. As I mentioned, linear free energy relationships are commonly used in synthetic chemistry to understand mechanism. And so the last story I'll tell you about is a collaboration between my group, members of Matt Sigmund's team, and also Heather Johnson, who was at Merck. You know, we recognized, right, that linear free energy relationships are used quite commonly in organic chemistry. And what they capture, right, is the continuous dependence of a given descriptor on reaction output, like relative rate. But in chemistry, it's not just about continuous relationships, right, between molecular structure and reactivity. And we were interested in whether we could build some tools that could, from a mechanistic perspective, allow us to capture non-continuous relationships, and in particular, reactivity cliffs. These are things that people describe quite a bit in reaction optimization. They find these, but it's sometimes hard to actually identify what the molecular feature is that's responsible for the reactivity cliff, and then basically connect that to mechanism. And what really led us to this is some chemistry that my group had worked on a number of years ago, wherein we were interested in a nickel catalyzed cross-coupling reaction of acetals to generate sp3 CC bonds. And when we were surveying this reaction, doing reaction optimization one variable at a time, what we found is that ligand identity, the phosphine identity, was critical. And ligands that were known that had been used for palladium and nickel previously only gave moderate yields. But what we found is that through some experimentation, that we could identify new ligands, in particular ones that had these 3,5 substituents on the arene of the alkyl phosphine that delivered high yield. And we have a tradition in my group of naming ligands ridiculous things, and so we've named these ligands after dinosaurs. So the question was why, right? Why are these ligands so effective? And early sort of evaluation from Kevin, suggested that maybe, right, the steric, the size of these ligands mattered. And what we found is that if you describe these ligands in terms of their cone angle, a classic descriptor for size of a phosphine, you can see that there seems to be a continuous trend, right, of the larger the cone angle, the better the yield. However, this broke down quite quickly, and what we found is there are a whole slew of other phosphine ligands that we had looked at that had, right, cone angles that either were larger or smaller, but didn't react. And so what it suggested to us is that there was a descriptor of a phosphine that we hadn't identified that was important, right, for reaction success, but we didn't know what that was. And so that's what prompted us to collaborate with the Sigmund team. Matt's lab was in the process of building a library of phosphine ligand descriptors, which they've named Kraken. And this was in collaboration with Ilana Spiroguzic's team, led by Toby Gensch and Gabe Gomez. And what was really important about, I think, their library, besides its size, it's sort of capturing all known commercial phosphines plus a virtual library, is that it was describing the conformations of those phosphines and featurizing sort of multiple conformations, such that you could get not just the Boltzmann average feature set, but the minimum, maximum value, as well as lowest energy conformer values. So we wanted to apply this to our data. And so I'll show you a few of these plots where we're looking at yield as a function of one of the features, in this case, cone angle. And each of the dots is a different phosphine that are color coded as shown sort of down here. The purple ones are our dinosaur ligands. the case of cone angle, you don't see a relationship here between cone angle and yield. But what was really interesting is as we looked at some of the buried volume features, in particular, this VBUR min. And what min describes here is the smallest buried volume that a phosphine will adopt within sort of an energetic range. What we see is sort of a quite sort of dramatic cliff where phosphine ligands that have buried volumes less than about 32% can be active, but those that have buried VBUR mins that are greater than that 32% cutoff are all inactive. To see whether this was something sort of that was, common to not just this particular reaction, we did HTE with a range of other reactions, in particular sp2 couplings with nickel. And I'll just show you a few of these data sets again, where we see this reactivity cliff with the VBUR min parameter, and we don't see it with the other sort of univariate parameters that we've investigated. And it turns out it's sort of common across different substrate classes for this reaction too. There's another example here. And so what this prompted is sort of a question of, is this mechanistically meaningful? What we came upon as a hypothesis is, we know in the phosphine palladium literature that speciation, whether a phosphine can bind 2-to-1 or 1-to-1, is critical to reaction outcome. And we wondered if this reactivity cliff, in the case of nickel, was a readout of whether a phosphine could bind 2-to-1, which would be sort of below that cliff, or 1-to-1, which would be above that cliff. So to test this, Sam and Julia in my lab designed an NMR experiment to read out whether phosphines can bind 2-to-1 or 1-to-1 to nickel. I'm showing here ligation state, so 2-to-1, 1-to-1. In the case of cone angle, you see no relationship. But interestingly, when you go to VBUR min, you do see that this actually featured specifically describes or reads out whether a phosphine can bind 2-to-1 to 1-to-1. And what was really exciting to us is that this cliff at the around 32% is exactly what we see sort of in the reactivity data as well as the speciation data. And just showing that to you here. So what this allows us to do is both make a mechanistic hypothesis that ligands need to be able to bind 2-to-1 to be active in these cross-coupling reactions, but it also allows us to draw on paper, completely new phosphine ligands, in this case, this very, very, very large phosphine, if you were to describe it based off of its cone angle. It's one of the largest cone angles known. But the VBUR min value is below 32% and allows us to make a prediction that it actually should bind 2-to-1 to nickel and potentially be active in catalysis. And that's exactly what we see, is that the phosphine can in fact bind 2-to-1 to nickel, and it is active in catalysis, sort of shown sort of right there. All right, so I know I'm running a little bit over, so I want to wrap up here, but just to give you a sense of why VBUR min is important relative to the other types of steric features that we examined. And I think the most important sort of case study to think about is sort of conformationally flexible ligands like triacetylphosphine. So the Boltzmann average VBUR of this is 39%, which is pretty sort of similar to the lowest energy VBUR. But when a phosphine binds to the metal center, right, it can potentially reorganize if it's sort of energetically feasible. And so in this case, we've gotten a crystal structure of the nickel complex bound to 2 trisabutylphosphines. And if we compute the buried volume of the phosphines bound, what we see is that their buried volume is very similar to the minimum buried volume of the library, so the Kraken features. And so we think it's the physically more meaningful description of a phosphine size, especially one that is conformationally flexible, that allows us to sort of read out ligation state when binding to a metal. So just I'll finish here to say that we've sort of formalized this threshold or reactivity workflow. It's a super simple, right, single node decision tree that can be used to find these reactivity cliffs and scan a list of features And when we do this, we see again, sort of in a formalized way, that VBER-min is the best, most effective descriptor in classifying these data sets. And then the last thing I'll end on here is that speciation is also important in other metal catalyzed cross couplings. And indeed, with palladium catalysis, we see that VBER-min does deliver thresholds just like we'd expect, and we see with nickel. Interestingly, you tend to see that phosphines, right, that can bind 1-to-1 are active, whereas those that bind 2-to-1 are inactive, consistent with what we know mechanistically for palladium catalyzed cross coupling, and except for cases that have been mechanistically studied and shown that 2-to-1 binding is important. So we've surveyed a range of data sets now for both nickel and palladium, where we see these thresholds that can allow you to make mechanistic inferences about the importance of speciation in nickel and palladium catalysis. We've also seen data sets where we don't see that threshold, and actually that also is informative in suggesting maybe that, for example, various nanoparticles are sort of catalyzing the reaction, not homogeneous catalysts. All right, so I will end there. I apologize for going over a little bit, but hopefully I've given you a sense of some of the data-driven tools that my group uses in the course of reaction development and catalyst design. I work with a fantastic group of students who I've named throughout this. I'll just do want to point out that many of our studies have been funded by a new center funded by the NSF called Computer Assisted Synthesis, led by Olaf Wiest. And so I'm very grateful for that funding, and I'd be happy to take any questions.
Thank you, Abby. Really fantastic. This was an amazing journey across different topics, and we do have a few questions. And also, I'm also reminding people that if a few more come to your mind, feel free to post them in the chat, ping Marie-Francine. Thank you so much. I'll start with the very first question. So let me just go through that one. So this was related to the Bayesian optimization. So one of the things that I'll just read the question without interpreting that. So basically, for the BO work, you showed that the performance of human experts' intuition led to higher yields while potentially having big impacts due to bias. Any thought on how one could try to quantify this bias in a more general way, and how do you think this will be important for the future of organic chemistry?
It's a great question. We've talked a lot about it. There was one beautiful study in the area of material science that sort of tried to capture this idea of sort of human bias in sort of experimental work. How do you capture? I mean, we've thought about if we could sort of apply our game in a more general sense to capture sort of decision making that happens in the course of reaction optimization or sort of other things, and then potentially use that to sort of learn about sort of priors that we could use in the course of for our Bayesian optimization work, for example. I would love to be able to do it. I don't know quite how to, and I think we also would very much need to get sort of experts to help us with the game design, since this was very rudimentary, right? And we didn't incentivize it, and so I'm not sure that people spent much time in the process of playing the game. But I do think it's a really interesting question. We have done, this is the really simple thing of initiating the optimizer using the chemist's initial decisions, right, for the first 5 conditions to run. As you might expect, that actually leads to poor performance for the optimizer, because right, those selections are likely sort of fairly localized in the chemical space, whereas right, rate of learning for the optimizer is improved if right the first 5 conditions are spread, right, and quite diverse. So that doesn't help, but really what we'd like to be able to capture is some of the the incremental decisions that sort of chemists make, where that intuition helps them in sort of the early stage of the optimizer campaign, because in those early stages, right, the response surface model is poor, right? And in terms of, they're very high uncertainty and very poor sort of predicted sort of mean.
And maybe if this is just a question that I'm adding on top of this one. Based on your experience, how do people react when they are confronted with this bias? I mean, I think this is an interesting sociological, chemical.
It is. Yeah. No, I mean, on the one hand, I feel like, when I've brought it up to chemists or people in my group, they get it, right? I mean, it's not too surprising. The thing that, related to that I've had a hard time with even within our group is, having students use the optimizer, right? It's sort of seeding decision-making, and they don't want to do that, right? Or at least they want to understand why they're being told to run, right, this experiment that to them, right, it's like this ligand will never work. So we've been trying to, with our new version, have at least sort of visualizations of the uncertainty of the response surface model, and predictions, mapped out so that, you can sort of infer a little bit, all right, they're telling me to run this experiment, which I think is ridiculous because, right, it's an area of high uncertainty. And because of that, I'm okay doing it, even though my intuition would say, right, it's not going to help, in actually getting to that high yielding set of conditions. So at least by giving them that information, I feel like, my students are a little bit more willing to sort of run some of those experiments.
No, thanks. This is really, as I said, an interesting experiment. Social, chemical.
I agree. I agree.
I hand it over to Marie-Francine, right?
Yes, perfect. So I will ask next question. So in the Bayesian reaction optimization study, you showed that you knew the ground truth beforehand from the HTE data. How feasible do you think this is for cases where one wants to apply it if the ground truth is not known and one wants to do it as per the algorithm recommendations?
Yeah, so I mean, that's what we are doing now, right? Is the case where, when we've been testing the algorithm, it's been, we've wanted to have that ground truth, right? So that we can sort of make statistical sort of, comparisons and assessments. And, from what we've seen, as long, as, the search space that you identify isn't huge, right? If you're dealing with, hundreds of thousands of experiments or smaller. From what we've looked at, we think that you can run, right, EDBO and within, right, 50 to 100 experiments identify what the global maximum is within that data set. Now, of course, we've not tried it on every single reaction type to, be confident, but, part of the beauty of, I think, using the algorithm is you do get to see how the uncertainties of the model and the predictions evolve, right? And presumably if the uncertainties are relatively low and stable, you can sort of figure out when to stop, right? All right, if I haven't reached a satisfactory result, but the uncertainties of the model are low, all right, I need to redefine my search space. And I think that's really useful. I mean, from my perspective in my group, knowing when to stop, right? An optimization campaign is hard, right? So we have this hypothesis, we think it should work, we've got a result, right? It might not be optimal. And so there's this, inclination to keep trying, right? But to know that, all right, we need to reevaluate and have sort of a data-driven, approach to that is really useful. So we don't have, hard, data to say, as long as it's, right, a certain size search space and you'll find the result within, 100 experiments. But I think, as we start to use it, we'll learn more and improve upon it. There's also this question of, we've not tried it in a case, although we're trying, now doing so where you're starting at like 1% yield or like barely, on the board, and that's another, right, challenge that I don't know if we'll be able to sort of use it as we have it right now to do that. And there's a whole other question of how do you get from the 0% to the 1%, and we're interested in that, but I don't think this is the tool for that.
Thank you. We have a further question on uncertainty. So regarding the UMAP maps, how much of the uncertainty is in the representative of a cluster, or in the representative of a cluster is due to hidden features? How do we account for non-explicit features and improve cluster exploration?
That's a great question. Yeah. So in the case that I showed you with aryl bromides, we're using DFT featurization for both. So we use molecular properties from DFT, like HOMO-LUMO energy, as well as atomic features that capture just the C6 carbons of the aryl bromide and the bromine. So when you have a decorated aryl bromide with functional groups, the featurization approach we're using right now doesn't actually capture those functional groups. It's just capturing the impact of the functional groups on the C6 core. And we think that's partly why this approach is only very good, effective at the internal sort of reactivity description rather than sort of the external impact. So in principle, some sort of sort of combined featurization approach that's something like fingerprints or a graph-based representation in combination with DFT featurization might be helpful there. And I know that like Connor Coley has had some success using sort of these combinations of featurizations for some predictive models and selectivity. So we haven't tried that, but I think something like that could be effective. But I think it's an important next step, and especially as we move away from, aryl bromides are a safe and easy place to start because there is significant sort of overlap between within the substrate class. But we've tried to apply this to heteroaryl bromides, just a small leap, and then you really only from atomic features can, if you're doing DFT, can capture the carbon and the bromine, of the heteroaryl bromide, because you move from pyridines to pyrimidines to, you don't have any other consistent atoms. So I think, it's really something we're starting to do as we sort of jump into new synthetic methods in my group to think about. We've only scratched the surface here.
So we have a few more questions and 8 minutes. Take a little bit of breath, water, just a few seconds of pause, but I will start slowly reading the next one, which is somehow related to the one that Lauren was just asking, because this question is asking, the work on the phosphine is really impressive. Thank you so much for the beautiful presentation. How do we generalize from phosphine to other systems, and how do we systematically come up with protocols that allow to identify the relevant features for those systems?
That's
to a certain extent, I think you mentioned some ideas already in the previous question, but I mean.
It's a great question. We are so one approach I'd say is like, the one that Sigmund and Spiraguzic did was, build these databases, at least for, common ligands, and I know that there's efforts making, a similar database for bisphosphines based off of some of my group's interests. We're starting to, with Matt Sigmund and Sarah Reisman, build one for nitrogen-based ligands. It starts to become more complex when you don't have a monodentate phosphine, but bidentate or tridentate ligands, and comparing between them and ones that are C2 symmetric and not C2 symmetric. So we don't have answers yet, but so one is through this database approach. The other is if you have more data, there's been lots of successes outside of my own group's work using other types of featurizations that are a lot less expensive than DFT based. And there the question is, can you get mechanistic, right, inference from those sorts of models. And I think that there will be the possibility of doing so, you know, as long as we've got good quality and a lot of data.
That's true.
That's a big ask, though.
Thanks.
Good. Then the next question starts with again, thank you for this beautiful talk. So the person asking was wondering about the last case study. So you showed an analysis looking only at one property at a time to find a descriptor. Wouldn't a combination of multiple descriptors be very likely? And what are the problems in looking at multiple descriptors at a time?
Yeah, so that's a great question. Yeah, so in this case, we were just sort of scanning univariate sort of single node decision trees. But clearly what you can see is in those plots I showed, you know, what, you know, these cliffs show you is that, you know, for ligands that, you know, are, you know, in the confusion matrix are sort of predicted, you know, in the active, right, quadrant. Some, right, still can be, right, very low performers and some are very high performers. So all this is telling you is that, right, that ligand can be active, you know, in catalysis rather than that it will be active. And so there's clearly other features there that govern the relative activity. And certainly one of them is electronic, we know. And so what we've started to do, we initially started by trying to do multivariate, some sort of modeling of these data sets, and we're not very effective. But what we have found subsequently, and I think other groups have found this too, is that if you apply these sorts of decision trees, it helps you in a data-driven way divide data sets such that you can then more effectively model, right, the quadrant of, you know, ligands that are sort of predicted to be active or have the possibility of being active, and doing multivariate or some sort of, right, algorithm on that section of the data set. And so we've had some success there, whereas we didn't have success initially trying to globally model that data. And so that's where we're going with this. And certainly the goal, you know, the big picture at the very beginning was to try and understand why these particular phosphines were so effective. We have one answer to that, which is that they bind 2-to-1 to nickel, which is actually was a huge surprise. We had thought that they were so big that they were only going to bind 1-to-1, but then still don't know why, you know, that ligand is better than other ligands that bind 2-to-1.
Thank you. Oh, sorry, Theo.
Go ahead.
Okay.
I was just saying nice.
Yeah, so I think we have time for one, maybe two final questions. So this one goes back to the Bayesian optimization. So how would you know when to stop a Bayesian optimization? For instance, for some of the reactions shown, the optimization stops at around 70%, while some were optimized until 100% yield. Could the 70% reactions eventually be further optimized to 100%?
Hopefully, you know, from a chemist perspective, you know, so what we've done, so in our original application of the Bayesian optimization, right, the chemist defines the search space, right? Says, I want to evaluate concentration, temperature, ligand. Here are the ligands. And you sort of were, it was static. There was no way of adding, you know, additional sort of experiments to that search space. But in the newest version, you can sort of on the fly sort of change the search space. And so I think our idea, and we're trying to sort of test this in practice, is that you define the search space, you run the optimizer. As you start to see, you know, if something does taper off at 70% yield, for example, you know, and the uncertainties of their, you know, of the response surface model are low, and you can look at all of the predictions for that entire search space and see that it's not predicting anything sort of higher, then you have the opportunity of expanding the search space, whether that's changing, you know, adding sort of additional ligands, or, you know, other variables to try and sort of get you above, right, that sort of plateau that you observed. Now, what we'd love to be able to do, haven't done yet, is, right, have some sort of generative model, built in for something like ligands, right? To select sort of new ligands to try. That would be great. We haven't done it, but that would also be a potential solution to this.
That would be
great. Thank you.
No, thanks, Lauren. And I wanted just to connect, saying that these would be very interesting topics for discussing cross-Atlantic collaboration.
Absolutely.
I think it would
love to.
So last questions before we wrap up. Let me just go through that. So how did you design the Dynophos ligands? Are they result from a computational calculations telling you that this structure would have had a low bar mean, or did you design on paper and then calculated the bar mean in a sort of trial and error way?
Yeah, it's a great question. There was no computational design associated with that project initially. It was, both really simple, but I think on the student's part, one of those leaps, it was not my, my idea. And I think the student was sort of brilliant in this. He basically was looking at, a whole range of phosphines that, are used commonly. And really quite simply recognized that there are many examples of orthosubstitution that sort of fall into the Buchwald ligand category that are used commonly for phosphines, and parasubstitution that are used to modulate electronics. But there was, and he didn't do any sort of systematic, visualization of this, but just sort of eyeballed it and recognized that 3 substitution, metasubstitution was under sort of explored in this space. In fact, I think there was only 1 or 2 monodente phosphines that were commercial, at least, that had metasubstitution. There was one called Drufos that had been previously published. And so, that was part of it. It was like, all right, this is chemical space that hasn't been sort of examined. And then the second, was more hypothesis driven, was that with palladium, the best ligands are orthosubstituted, but nickel is smaller. So why don't we move that substituent 1 position away, and maybe that'll help. So yeah, no computational design at all. That was really the 2 criteria that led us to these ligands. And then the data-driven part was all sort of a second project after, that we had discovered these ligands. But certainly we'd like to be able to start using these to get to even more effective, more reactive ligands.
Okay, we are already 2 minutes over time, and we like to be Swiss, really on time. So I know that there were a couple of other questions that were coming right now in the chat. Apologize for everybody. I would like to thank again Abby for having spent with us some time, and everybody for having participated with questions. So thank you so much. I leave it to
I'll say, feel free to email me if I didn't get a chance to address the question. I'd be more than happy to do so by email.
Absolutely. And Marie-Francine, to you, Lauren.
Yes, also a big thank you from our side. It really was a pleasure to have you here. And yeah, we look forward to further exchanges then.
Absolutely. Thank you so much for the invitation.
No problem. I would like only to say, for people of the NCCR, maybe we can finish our cup of coffee. I'm a caffeinated person until 7 in the evening in the other Zoom room. And if you have a few things to ask to Abby, she will be there for 15 minutes. So let's be considerate over time. See you there. Thank you so much, all the others, for having joined.
Yeah, thank you so much. Bye.
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 Thank you.